So, in today's presentation, we are going to discuss the following topics: definition, its significance, renal physiology, etiology and pathophysiology, qualitative and quantitative estimation of proteinuria, symptoms, treatment, and management. So, now I'd like to pass the mic to Rajushi.
A very good morning to all my respected professors and dear friends. Myself, Rajoshi Raut, having roll number 92. Now, I'll start the discussion with the definition of proteinuria. So, what is this proteinuria? Proteinuria is a medical condition characterized by abnormal amount of protein. So, if this is the abnormal amount, what's the normal value? The normal value lies below 150 milligram per day. Hello, and why this proteinuria takes place? The in normal condition, the healthy kidneys, our healthy kidneys act as filters that allow the waste products to pass into the urine while keeping the essential protein as filtrate, which can get into the bloodstream. But in proteinuria, this filtration system gets damaged. Now let's move on to the next slide. What is this albuminuria? Albuminuria is the condition where excessive amount of albumin is present in urine, and in normal condition this range lies below 30 milligram per day. Here is a picture where we can compare a normal kidney with a kidney affected with albuminuria. So here we can see these are the albumin molecules which are already filtered in the urine, but in normal condition these are absent. So why this proteinuria is significant? The proteinuria is basically an indicator of early kidney disease. It can indicate both acute and chronic kidney disease. It is important for the evaluation of management of chronic kidney disease. It indicates glomerular as well as tubular damage, and it's also a strong predictor of adverse cardiovascular and kidney events. Now let's move on to the next slide. So for understanding the impairment of kidneys, we need to understand in normal condition how kidneys act as the urine filter. So let's get a glimpse of the renal physiology. So first of all, nephron is the functional and structural unit of kidney, and the parts of nephron are glomerulus Bowman's capsule, the proximal convoluted tubule, loop of Henle, and distal convoluted tubule and collecting duct. So this urine formation takes place in 3 steps. The first step is glomerular filtration, which occurs between the glomerulus and the Bowman's capsule. The glomerular filtration allows smaller solute particles to pass on, but it usually restricts the passing of the plasma proteins from entering the urine, and keep the protein as filtrate, which is called glomerular filtrate. The next step is tubular reabsorption. It occurs in the proximal convoluted tubule, loop of Henle, and distal convoluted tubule. Substances like glucose, amino acid, water, sodium, and other ions are reabsorbed from the filtrate back into the blood. And in the final step, the tubular secretion, which occurs mainly in distal convoluted tubule and collecting duct. Here, hydrogen ions, potassium and ammonia, these are secreted, and this step is crucial for maintenance of electrolyte balance as well as maintenance of blood pH. And when all these steps are completed, the remaining fluid can be called as urine. So my part comes to conclusion here. Now I'll hand it over to Rajendra.
Good morning, everyone, and good morning, respected sirs. My name is Rajanna Mondol, and my roll number is 91. And I will be continuing from this slide. we know that the some normal protein present in urine, and this proteins comes from the two parts. From one is the plasma proteins and one is the tubular cells. From the plasma proteins, we know that there is an albumin which is present in 20% and others is 80%. There is some transfer in alpha 1 and the microglobulins. From the renal tubular epithelial cell or the renal cells, we know that here is a protein called tam protein, which is a very important role in the urine, and also known as uromodulin. It plays an important role like immune defense and preventing renal kidney stones. And the next slide is the etiology, where we discuss some physiological and the pathological causes. In physiological causes is the transient proteinuria. It is occurs in the healthy and the normal kidneys, which is a temporary and the reversible process. Usually, a small amount of protein released, but no structural kidney damage. And some other physiological causes like fever, stress, molecular body builds, heavy exercise, and the pregnancy. And some other causes is the orthostatic proteinuria, and the orthostatic proteinuria cause like in the upright position protein urinary protein secretion may be increased to 10-fold up 1000 mg per day, and no protein excreted in the urine while laying down, so the proteinuria in the morning. Common in the children, it is very common in the children and the adolescents. And here we discuss some pathological causes. In the normal condition, normal condition glomerular filtration is occurs where the proteins are present in the urine. In the overflow condition, there are some high amount of protein which is present in the urine. Like Benzos proteinuria. In the overflow condition, Benzos proteinuria occurs. And the next part is the glomerular filtration, where the glomerular basement membrane can ruptured, and some excess amount of proteins are present in the urine. The tubular secretion, glomerular filtration is normal, but some proximal tubules have failed to absorb these proteins, and the secreted proteinuria, some renal tubules fail to absorb these proteins, and which is present in the urine. And the glomerular causes, we know that the glomerular has three types of this parts, like endothelial cell membrane, and secondly is the glomerular basement membrane, and third part is the podocyte cell. When some damage occurs in the glomerular endothelial cell, it causes the glomerular basement membrane become leaky, and the albumin like proteins are secreted from here, and the podocyte cell can be detached. So plasma proteins cross the damaged glomerular filtration barrier, and which is secreted into the urine. And in the glomerular causes, there are some disease like glomerular nephritis, diabetic nephropathy, lupus nephritis, which is causes and which leads to the nephrotic syndrome. Then is the tubular cause. Normally, tubules of the nephron reclaims low molecular proteins, and proximal tubules have failed to absorb this kind of the proteins, and whereas the range is greater than less than 2 gram per day, and it causes like disease like acute tubular necrosis, Fanconi syndrome, heart-lung disease, toxic, and the medication. Now we will be passing this mic to Rijorita for continuing this PPT.
Now we will also go through the pathophysiological causes of proteinuria, which is one is the overflow of proteinuria, which is the increased plasma concentration of low molecular weights is normally filtered out by the glomerulus, which is that the proteins filtered out is more than the proteins being reabsorbed. So this results in excess amount of proteins in the urine. The causes can be multiple myeloma, it can be excess myoglobin and also excess hemoglobin. The post-renal causes can be any obstruction in the renal pelvis, the ureter, the bladder, the prostate, the urethra. The examples can be the urinary tract infections, bladder tumor, and self-inflammation in the urinary tract. Now, some other clinical causes can be the renal trauma, which is physical injury to the kidney. It damages the kidney's glomerulus, and which results.
Warning bell.
which results in the leakage of the protein in the urine. Post-renal transplant proteinuria, this is usually seen when there is a kidney transplant. It can be due to acute rejection or where the immune system attacks the transplanted kidneys. It can be due to any recurrent occurring diseases or due to any immunosuppressive drugs. Now, the qualitative test for proteinuria are four types, which is heat and acetic acid test, the sulfur-salicylic acid test, the Heller's nitric acid test, and the dipsticks test. We will go through the heat and acetic acid test. The principle is that with the application of heat, the protein structure usually gets denatured, which means that the protein forms a white semi-solid coagulant, which is irreversible in nature. Now, heating the alkaline medium, it usually causes the urine to precipitate out some calcium phosphate ions, which is actually reversible in nature. But when we add acetic acid, 10% acetic acid, we can confirm that the urine is having protein or it is having phosphates. The procedure is we fill the 2/3 of the test tube with urine. We heat the 1/3 of the tube and we see a white coagulant formation, and then we add acetic acid. We can confirm two observations from this. The first observation is that the coagulation formation, when we add acetic acid, the coagulant is remaining even after that. It means protein is present in the urine. And if the coagulant disappears, it means that phosphate is present.
Next, qualitative taste is sulfur salicylic acid taste. So we basically mix 3% sulfur salicylic acid to the urine sample. This acid normally lowers the pH and this denatures the protein, and as a result, the protein precipitates in the urine, causing turbidity or cloudiness. One clinical advantage of this test is we can detect all the types of proteins. Like here we can see after we observe the test tube against a dark background, when there is no cloudiness, which means the proteins are absent in the urine. When the cloudiness is in a very faint, we have trace amounts of protein, and like in the last test tube we can see the cloudiness is very dense, which means the large amounts of protein are present in the urine. So greater the turbidity, greater is the amount of protein present in the urine. The next taste is Heller's nitric acid taste. We add 2 to 3 ml of concentrated nitric acid in a test tube, and we add the urine along the side of the test tube. Nitric acid denatures the proteins, and as a result, precipitation forms at the junction of the two liquids, and a white ring is formed. When the white ring is absent, which means the protein it is absent in the urine. Now we would look at the quantitative taste, the 24-hour urine protein test. In this test, we normally collect the urine sample for over 24 hours. The first morning urine is generally discarded. When it is less than 150 mg per day, it is normal. When it is 150 to 500 mg per day, we have mild proteinuria. Greater than 500 mg per day, moderate proteinuria, and if it is greater than 3500 mg per day, it is nephrotic range, which is severe proteinuria. The second test is spot urine protein creatinine ratio. We use a single urine sample and we compare it with creatinine. If it is less than 150 mg per gram, it is normal. 150 to 500 mg per gram, mild proteinuria. Greater than 500 mg per gram, severe proteinuria. The last test is spot urine albumin creatinine ratio. If it is less than 30 mg per gram, it is normal. 30 to 300 mg per gram, microalbuminuria. Greater than 300 mg per gram, it is macroalbuminuria. Now, I'm passing the mic to Rishi.
Good morning, everyone. I am Rishad Iqbal, roll number 95. I am going to discuss about symptoms of proteinuria and prevention and treatment. In early stages, proteinuria may not show any symptoms. Symptoms are usually seen in advanced stages. Symptoms are frothy, foamy urine. This is the most common symptom. Excess Excess protein decreases the surface tension in urine, which produces persistent foam. We can differentiate from normal cause by seeing the foam lasting longer than usual urine bubbles. Edema, swelling. This occurs when there is loss of plasma protein, especially albumin, leading to lower plasma oncotic pressure, causing accumulation of fluid in interstitial spaces, leading to edema. And the next point, weight gain, is also related to the previous one.
Anyway, overall it's a very good presentation. All of you have participated very well. Speech was very good. Delivery is also very good. Coverage of the topic is very good. And most beautiful thing I found is the way you have prepared the PowerPoint presentation. Excellent.
In one slide, somebody has shown that 3.5 gram per day. Who has presented that slide? 3.5 gram per day urine excretion. Yes. How will you get that per day, 3.5 gram per day? How will you estimate? Number one question. Number two, who has told microalbumin and macroalbumin? Yes, what is the difference between microalbumin, macroalbumin and albumin? What is the difference between among the three? Microalbumin, macroalbumin and albumin. Okay.
Good morning, teachers and fellow batchmates. I am Rishika Sahu, roll number 96, and today in this presentation, we will discuss the definition, classification, structures, and functions of immunoglobulins. So, let's get started. Immunoglobulins, also known as antibodies, are special proteins formed by plasma cells and which are formed by B lymphocytes. They are produced when a foreign body known as antigen, such as bacteria, viruses, or toxins, enters our body. The main function of immunoglobulin is to recognize the antigens and then bind with them and help them remove from the body. Therefore, they are an important part of humoral immunity, which protects us from infection. Now, let's move on to structure of immunoglobulin. As you can see, it is Y-shaped glycoprotein. There are 4 protein chains. It is made up of 4 protein chains, 2 heavy chains and 2 light chains. The chains are connected via disulfide bonds, and the the tip of the Y is known as the variable region where the antigen binds, so it is known as the antigen binding site, and the remaining part is known as the constant region, which helps to activate immune response. The middle flexible part is known as the hinge region, which allows the antibody to bend and attach to the antigen easily. Now I would like to hand over the mic to Ruturaj.
Good morning, everyone. Now, talking about FAB and FC region. First of all, the two arms are termed as FAB region. Mainly, the upper two domains. The upper two domains are variable domains, which helps in binding the antigens. Same on this side. Rest of them are constant domains. The variable domain ensures that specific antigens are binded to the antibody. Each antibody have two FAB regions, so two antigens can be binded to the antibody. Now about FC region, the stem portion is the FC region, composed only of the constant domain, while FAB region contains both variable and constant domain. Does not have any antigen binding region, but it still plays a crucial role in mediating the immune response because it's responsible for activating the complement system, binding to FC receptors present on immune cells like macrophage and natural killer cells, and facilitating process such as opsonization, in which mainly IgG antibody covers the pathogen and let the phagocytes to ingest and destroy the phagocytes, pathogens. And it is also helpful in antibody dependent cell mediated cytotoxicity. Now about the classification of antibody. It is divided into 5 major classes. We can differentiate based on the heavy chains, but structurally we can easily differentiate. As we can see, the IgM has 5 polymers. It's pentamer, while IgA is a dimer. Sometimes it's a monomer too. Uh, IgE can be IgE can be differentiated as there are 4 constant domain on each sides. Well, while IgG and IgD contains 3 constant domains. To differentiate between IgG and IgD, we can look at the hinge. While IgG got shorter hinge, IgD have longer hinge. So the shorter hinge provides more stability. So IgG is more stable as compared to IgD. As we know that IgA sometimes uh, is also available as monomer, uh, but we can differentiate with IgG and IgD based on the hinge, as its the size is intermediate in between IgG and IgD. The talking about hinge is neither a part of FAB region or FC region, but it functionally takes part. Functionally, it helps or it take parts in FC region. Now, for the further explanation of IgG and IgA antibody, I would like to call Rudra.
Good morning, everyone. Now I will discuss immunoglobulin G. It is the most abundant immunoglobulin, and 75% of total serum, and molecular weight 150 kilodalton. Structure, it is the monomeric structure and Y-shaped, 2 heavy chain and 2 light chain. The chain are connected by disulfide bond, and it it's generally found in body serum, lymph fluid, various others body, and placenta also. Functions IgG, your immunoglobulin G can cross the placenta and also um, neutralize the bacteria, virus, will act as a opsonin and phagocytosis, produce secondary immune response, and also passive immunity to the fetus. Next, immunoglobulin A. It is the second most abundant immunoglobulin, 10 to 15%, and 10 to 15% of total serum. Molecular weight 160 kilodalton. Structure monomer in serum and dimer in secretion. It is also Y-shaped and 2 heavy chain and 2 light chain are connected by disulfide bond. Also found in serum, saliva, tears, colostrum, or breast milk, etc. Functions: provide mucosal immunity, provide passive immunity to infant through breast milk, and also neutralize bacteria, virus, and uh, toxin part also, and prevent attachment the microorganism. Thank you.
Good morning, everyone. Respected sir and ma'am. I will continue from here. IgM. IgM, it is also known as mu immunoglobulin because its its heavy chain represent as mu. It is largest immunoglobulin, also the heaviest immunoglobulin, because its weight is 900 kilodalton, and it is found throughout the bloodstream and the lymph fluid. It is, as seen, it is pentameric in structure, so it has 10 antigen binding site. It is also known as decavalent, as its valency has 10 antigen binding sites. It has high affinity because the antigens are 10 antigens can bind together, not together because of steric hindrance. It will unstable the structure. It is This is the J chain, which is joined by the 5 monomers with the disulfide bond. Now the functions of IgM, as it is the first immune response whenever a foreign particle or bacteria is shown. So it is the first response given by the body. Also, it has an excellent agglutination property because its structure is very large, and also it helps in RBC clotting. So as it is written, it is the earliest antibody created by the body during the primary response. Finding elevated IgM levels in the blood test typically indicates any very recent or active informations. It is due to its massive and pentameric structure. It excels at clumping foreign cells together, make it easier for the body to filter and remove them. IgD. IgD is also its concentration in the immunoglobulin total is less than 3%, and it is monomer in nature. Also, it is located on the mature B cell surface of the mature B cells. It is an antigen receptor, as its function is not specified, but it has a function along with IgM. It acts on beta cell, and it acts as beta cell receptor. Thank you.
Hello, I am Rupam Roy. Good morning, my respected teachers. I would like to explain IgE epsilon. It's an antibody which is actually produced by our immune system to our fight off our body from parasites, and it is found in mucous membrane, skin, and lungs. Also found attached to basophils and mast cells. And for the functions, it's fighting parasites. It helps recognize and expel parasitic worms and protozoa, like it's uh, activates eosinophil, which releases toxic proteins, uh, which helps to remove the parasites from our body. And the next point is allergic reactions. Sometimes it mistakenly recognize uh, stuff like pollen, food, and pet dander as allergens. That's why sometimes our body can show allergic reactions like swelling, itching. And the next point is immune activation. Ig binds to mast cells and basophils, and it releases chemicals like histamine, which causes vasodilation, bronchoconstriction, swelling sometimes. Uh, and now I'm gonna do the comparison of immunoglobulins. First, IgG, placental transfer, IgA mucosal transfer immunity, IgA primary response, IgD for beta cell receptor, IgE for allergy, and IgG is the most abundant among all, and IgE is the least abundant. The concentration of immunoglobulins in serum is in mg per dl, which is 1000 for IgG. For IgA, it's 200. For IgM, it's 120. IgD is 3, and IgE for 0.05. And sedimentation coefficient, it's respectively 7s or 115A, 195, 7s, and 8s. And according to molecular weight, it's in kilodalton, 150, 170, 900, 180, and 190. The structures are monomer, and for IgG, and for IgA it's monomer or dimer, and for IgM it's monomer or pentamer, for IgD it's monomer, and for IgE it's also monomer. And the heavy chain symbols are for IgA is gamma, for IgA is for alpha, and IgM is mu, and IgD is delta, and it's epsilon for IgE. And for opsonization, it's only seen in IgG, and the antigen receptor on beta cell it's only seen in IgM. I'd like to hand over the mic to Rishika.
Thank you, Rupam. And to conclude the presentation, I would like to talk about the summarized functions. For IgE, it is the most abundant in secondary response, opsonizes bacteria, making them easier to phagocytosize. Fixes complement, which enhances bacteria killing, neutralizes bacterial toxins and viruses, crosses the placenta. IgG is the only immunoglobulin which can cross the placenta. Secretory IgA prevents attachment of bacteria and viruses to mucous membranes, does not fix complement. IgM produces in the primary response to an antigen, fixes complement, does not cross the placenta. IgD is found on the surface of the B cells, where it acts as a receptor for the antigen. IgE mediates immediate hypersensitivity as it acts on the allergy part by causing release of mediators from mast cells and basophils upon exposure to antigen, which are allergens. It defends against worm infection by causing release of enzymes from eosinophils, does not fix the complement and main host defense against helminthic infections. Thank you.
Overall, good presentation. Thank you very much.
Good morning, everyone. The members of our group are myself, Shagun Gupta, Samiksha, Shamraghi, Shantanu, and Shuttujit. And our topic of discussion today is role of vitamins as coenzymes. Now, a vitamin is an organic compound which is required for proper metabolic functioning and overall health. A coenzyme is a molecule that binds to an enzyme to catalyze a specific biochemical reaction. Today we cover 9 vitamins. First of all, we start with vitamin B1, that is thiamine. It is a water-soluble vitamin which is required for carbohydrate metabolism and reactions that produce energy. Its active coenzyme form is thiamine pyrophosphate, which is formed when thiamine is combined with ATP. The type of reaction that TPP catalyzes is oxidative decarboxylation and transketolation reactions. Moving on to the actual reactions. In link reaction, pyruvate dehydrogenase converts pyruvate into acetyl-CoA. In TCA cycle, alpha-ketoglutarate dehydrogenase converts alpha-ketoglutarate into succinyl-CoA. In branch chain amino acid metabolism, branch chain alpha-keto acid dehydrogenase converts keto acid, alpha-keto acid into branch chain acyl-CoA derivatives, metabolizing amino acids such as valine, leucine, isoleucine, etc. In pentose phosphate pathway, transketolase converts xylose 5-phosphate and ribose 5-phosphate into glyceraldehyde 3-phosphate and cedoheptulose 7-phosphate. Moving on to the importance of thiamine. It helps in ATP production, pentose phosphate pathway, amino acid metabolism, and it is especially required in organs with high energy demand such as the heart, the brain, and the skeletal muscles. Moving on to vitamin B2, riboflavin. Now, riboflavin is a water-soluble vitamin and is a precursor of flavin coenzymes. Now, the word flavin comes from the Latin word flavus, which means yellow, that gives it its naturally occurring yellow color. The active coenzyme forms of riboflavin are FMN, which is flavin mononucleotide, and FAD, which is flavin-adenine dinucleotide. It is formed when riboflavin combines with ATP to give us FMN, and FMN then combines with adenosine monophosphate to give us FAD. Both of these take part in redox reactions by accepting and donating electrons. Moving on to the reactions, in electron transfer chain, FMN is required by the enzyme NADH dehydrogenase to convert NADH into NAD+. Coming to functions of FAD, in TCA cycle, succinate dehydrogenase converts succinate into fumarate. In beta oxidation, acyl-CoA dehydrogenase converts acyl-CoA in trans-delta 2 enol-CoA. It is the first step. Now, purine metabolism, xanthine oxidase converts hypoxanthine and xanthine into uric acid. And in antioxidant defense, glutathione reductase oxidizes glutathione and into reduced glutathione. Moving on to the metabolic importance, FMN and FAD help us in ATP production, electron transport, beta oxidation of fatty acids, and antioxidant defense. That is it with vitamin B1 and B2. Now I would pass on the mic to Samiksha to carry with vitamin B3. Thank you.
Good morning, everybody. I am Samiksha Gupta, role 102. Starting off with vitamin B3, niacin. Niacin and nicotinic acid are synonyms of each other. The active form is in the niacin is present in the form of niacinamide or nicotinamide. The coenzyme forms of niacin are NAD plus and NADP plus, that is nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. Speaking about some very basic differences between them, NAD plus is mostly involved in the catabolic reactions which produces energy, and the NADP plus is mostly involved in the anabolic reactions, also in the biosynthesis and the antioxidant defense mechanisms. The mechanism of their function, they are mainly involved in the oxidation reduction, that is redox reactions, and they function by carrying the electrons or the hydride ion in the metabolic reactions. As we can see, the NAD plus gets a hydride ion to get reduced to NADH, and similarly the NADP plus takes up the hydride ion to get reduced to NADPH and H plus. Speaking about the reactions of NAD plus and NADP plus. NAD plus, firstly the process is glycolysis. Here the enzyme involved that uses NAD plus is glyceraldehyde 3-phosphate dehydrogenase, which converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. Secondly, in the TCA cycle, isocitrate dehydrogenase, which converts isocitrate to alpha-ketoglutarate. Secondly, alpha-ketoglutarate dehydrogenase, which converts alpha-ketoglutarate to succinyl-CoA. And third, malate dehydrogenase, which converts malate to oxaloacetate. Next, we have oxidative phosphorylation. Here, the NADH donates the electrons to the complex 1 of ETC, where through the entire process of ETC, the ATP is generated. Also, one NADH can produce approximately 2.5 ATP. NAD plus also has a role in DNA repair. The enzyme involved here is PARP, poly-ADP ribose polymerase enzyme. Next, moving on to NADP plus, the main role of NADP plus is seen in HMP shunt pathway reactions, where glucose 6-phosphate is converted to 6-phosphogluconolactone by the enzyme glucose 6-phosphate dehydrogenase. Then the 6-phosphogluconate dehydrogenase converts 6-phosphogluconate to ribulose 5-phosphate. Next, in the fatty acid synthesis, the acyl intermediates are converted to a reduced form by the fatty acyl synthase. Then, lastly, in the antioxidant defense, the glutathione is converted to its reduced form, which protects the cells from the oxidative damage. Moving on to reactions where either NAD plus or NADP plus can be used. The most important reaction out of them is glutamate dehydrogenase reaction, where glutamate is converted to alpha-ketoglutarate. Moving on to vitamin B5 pantothenic acid. The active coenzyme form is coenzyme A. Coenzyme A has the presence of a sulfhydryl group in its terminal position,
That self-hydryl group is responsible for carrying the acyl groups and transferring it from the substrate to the product. Moving on to the reactions of vitamin B5 pantothenic acid. Firstly, we have pyruvate dehydrogenase complex enzyme dihydrolipoyl transacetylase, where pyruvate is converted to acetyl-CoA. Next, we have TCA cycle reaction citrate synthase. Here, the acetyl-CoA enters the TCA cycle by forming citrate on condensation with oxaloacetate and water. Next, we have fatty acid oxidation. Here, the fatty acid is oxidized to form acyl-CoA by acyl-CoA synthetase. Then, fatty acid synthesis. There we have the acetyl-CoA is converted to the malonyl-CoA by acetyl-CoA carboxylase enzyme. Next, ketogenesis, where the acetyl-CoA and the acetoacetyl-CoA is converted to HMG-CoA by the enzyme HMG-CoA synthase. Next, branched-chain ketoacid, alpha-ketoacid dehydrogenase complex. Here also the alpha-ketoacid is converted to the acyl-CoA form by the help of the coenzyme A. Then cholesterol synthesis. Here the HMG-CoA is converted to the mevalonate by the enzyme HMG-CoA reductase. And lastly, acetylation reactions. Here the acetyl-CoA with a particular amine substrate is converted to an acetylated substrate. Coenzyme A reactions are also there are also examples of coenzyme A like acetyl-CoA, succinyl-CoA, propanyl-CoA, etc. Thank you. Now moving on to Shamrat.
So now we'll move on with vitamin B6, that is pyridoxine. The active form is pyridoxal 5' phosphate, that is PLP, and the alternative active form is pyridoxamine 5' phosphate, that is PMP. Now this PLP is involved in amino acid metabolism. So amino acid metabolism, we see transamination and decarboxylation. In transamination, what happens is the PLP transfers an amino group from amino acid to keto acid. Now, as we can see in the given reaction, the amino acid, that is number 1, and keto acid 2, in the presence of transaminase and PLP, transfers into keto acid 1 and amino acid 2. Then we come to decarboxylation reactions, that it assists enzymes in removal of the carboxylic group, the carbon dioxide from amino acids. Next, it also helps in neurotransmitter synthesis. For example, serotonin from tryptophan. Then we have histamine from histidine, and we have dopamine and norepinephrine from tyrosine, and GABA, that is from glutamate. Now, it also helps in heme production, which is very important because the first and the rate limiting enzyme required to create heme, that is the LA synthesis, and PLP acts as a coenzyme for that particular enzyme. Now, we move on to ease it out. We have a chart here. We have the pathway for transamination that we have almost discussed previously. It helps to convert the amino acid and alpha-ketoglutarate to keto acid and glutamate. Then we have deamination, that is serine and threonine dehydratase. It converts serine to pyruvate. And for heme synthesis, we have LA synthase, which converts glycine and succinyl coenzyme A to LA. Now we move on to vitamin B7, that is biotin. So, in biotin, we have this is the active and the naturally occurring form of vitamin B7, that is D-biotin. Now, we move on to this particular part where the carboxylation of biotin, where this urido ring of biotin, it traps and binds CO2 in an ATP-dependent reaction. And for transfer of substrate, we have this particular this biotin lysine conjugate swings the activated CO2 over the target molecule's active site, and thus it transfers it to the substrate. Now we have a chart again. So in gluconeogenesis, with the help of enzyme pyruvate carboxylase, it transfers pyruvate to oxaloacetate. And fatty acid synthesis, we have acetyl coenzyme A carboxylase, and acetyl coenzyme A is converted into malonyl coenzyme A. Similarly, in amino acid metabolism, we also get the use of biotin, and in leucine metabolism, we also get biotin. So, due to shortage of time, I'm not explaining these parts. We move on to the next vitamin, that is vitamin B9.
So,
Thank you, Samragni. Now I will continue from B9. So, B9 is also known as folate in natural form and has synthetic form known as folic acid. Okay. Then it is a water-soluble B complex vitamin essential for DNA synthesis, cell growth, and cell division. Now, the folic acid structure, it consists of three distinct chemical moiety. That is, first of all, the pteridine ring. It is also known as 2-amino 4-hydroxy pteridine ring. Then the para-amino benzoic acid and the glutamic acid residue. The active form of the folic acid is tetrahydrofolate. The one carbon metabolism, it is used in the one carbon metabolism. In this metabolism, the tetrahydrofolic acid carries one carbon unit and transfers it from one molecule to another except CO2. One carbon unit which it carries are methyl group, methylene, methinyl, formyl, and formamino. The major sources of one carbon unit is serine, and then the glycine, histidine, tryptophan, and formate. These are the pathways which the tetrahydrofolate derivative forms helps to conduct. First, the N10 formyl tetrahydrofolate in purine nucleotide synthesis enzyme used is GAR transformylase or AICAR transformylase, that is glycinamide ribonucleotide or aminoimidazole carboxamide ribonucleotide transformylase, which converts the GAR and AICAR into formyl GAR, formyl AICAR, and convert it into IMP, that is inositol monophosphate, which is a purine nucleotide. In next pathway, that is in pyrimidine synthesis, the N5N10 methylene tetrahydrofolate derivative is used, in which the enzyme thymidylate synthase converts the DUMP, that is deoxyuridylate, converts into DTMP, that is thymidylate. And then in pathway of conversion of serine to glycine, the tetrahydrofolate is used, in which the enzyme serine hydroxymethyl transferase converts the serine into glycine plus N5N10 methylene tetrahydrofolate. In histidine metabolism, enzyme form amino transferase converts the form amino glutamic acid into glutamate plus N5 form amino tetrahydrofolate. And in methionine synthesis, the N5 methyl tetrahydrofolate derivative is used, in which the enzyme methionine synthase converts the homocysteine plus N5 methyl tetrahydrofolate into methionine plus tetrahydrofolate. Now, vitamin B12, which is also known as cobalamin. Vitamin B12, it is a water-soluble vitamin. It is synthesized only by microorganisms. Stored mainly in the liver and essential for DNA synthesis and helps in neural regulation, nerve function, and it has two active coenzyme form. One is the methylcobalamin, another is the adenosyl cobalamin, which is 5' deoxyadenosylcobalamin. cobalamin. So first, the methylcobalamin, it is used in various pathways like regeneration of tetrahydrofolate from N5 methyl tetrahydrofolate, then DNA synthesis, which is indirectly by regenerating tetrahydrofolate for nucleotide synthesis, and then conversion of homocysteine to methionine, which has the common reaction in which the enzyme methionine synthase converts the N5 methyl tetrahydrofolate plus homocysteine into tetrahydrofolate plus N5 tetrahydrofolate plus methionine. And another pathway in which the methylcobalamin is used, that is in synthesis of S-adenosyl methionine, which is an universal methyl donor, in which the enzyme methionine adenylyl transferase MAT converts the methionine plus ATP into S-adenosyl methionine plus inorganic phosphate plus pyrophosphate. The benefit of this coenzyme is it is critically for It is mainly useful for.
So just quickly, I say it's a very long presentation, but anyway, your preparation was good and presentation was good. Very good.
See, one thing I would like to say that it is a really academic dense presentation, there is no doubt about it. It is very difficult to remember all the functions against each enzyme, coenzymes. Okay? So try to remember at least two. And suppose from the presentation what I have seen, if I set an MCQ, perhaps none of us you will be able to answer properly. Because so many functions it is difficult to remember. Try to remember at least two of each vitamin so that you can write something on the examination on the answer sheet, or you can tell before the examiner. At least two common, at least try to remember. It is very difficult. You cannot remember all these things and you cannot produce it also. High chance of committing mistake. Okay? But all the reactions almost have been covered. There is no doubt about it. They have collected from good sources. There is no doubt about it.
Tejashwini, roll number 144, along with my fellow teammates. She is Tanvi Kumar, roll number 143, and he is Tamogna, roll number 141. And we are here to present our topic, which is on polymerase chain reaction. What is exactly polymerase chain reaction? Polymerase chain reaction is a molecular biology technique used to amplify a small segment of DNA in vitro. It works by repeating cycles of heating and cooling, allowing the DNA to be copied many times with high specificity. In today's time, PCR is widely used in medicine, diagnostic, forensic science, genetics, and research. Now look at the history of the polymerase chain reaction, which is invented by Karen Mullins in 1983, revolutionizing the molecular biology by enabling the rapid amplification of specific DNA sequence. Who also introduced Taq polymerase, which is a heat-stable enzyme which obtained from Thermus aquaticus, made PCR faster, automated, and more efficient. Today, PCR is essential source in medical diagnostic, genetic testing, forensic science, biotechnology, and biomedical research. Now, look at the principle of polymerase chain reaction. This process mainly relies on the specific primers, a thermostable DNA polymerase, which is a Taq polymerase, deoxynucleotide triphosphate DNTPs, and repeated cycles of temperature changes. During each PCR cycle, the target DNA is approximately doubled, resulting in exponential amplification of the desired DNA fragment. This high sensitivity and specificity made PCR an indispensable technique in medical diagnostic, genetic analysis, forensic science, biotech, and biochemical research. Now let's see the requirements of PCR. First, we need the template DNA. Contains the target DNA sequence to be amplified. Secondly, we need the primers, short DNA oligonucleotides that define the region of amplified. Next, we need thermostable DNA polymerase enzyme, which is a Taq polymerase that synthesizes new DNA strands at high temperature. Next, we need DNTPs, which is deoxynucleotide triphosphate required for DNA synthesis. And next, reaction buffers maintain the optimal pH and ionic environment for the enzyme activity. Next, we need some enzymes, which is a magnesium ions, which is an essential cofactor that enables DNA polymerase to function efficiently. And lastly, we need the thermocycle, which is an instrument that precisely controls the temperature changes required for the PCR. And for the next, I would like to hand over the mic to Tamogna.
Good morning everyone, I am Tamogna Ghosh and I will be presenting the steps and types of PCR. So PCR is the one of the most revolutionary technology in molecular biology. If we get a single blood strain from a crime scene or a sample of a viral patient, we can use this technology to make billions of copies of the DNA templates, and then we can study and diagnose it later. So the PCR usually have three steps: denaturation, annihilation, and extension. So in the first step, in the denaturation, the temperature is increased to around 90 to 98 degrees Celsius, in which the DNA will split into two strands. This is important because until and unless the DNA is split, the polymerase cannot act upon it. Then we will go to the second step, that is the annihilation. In this step, the temperature is brought down to around 40 to 60 degrees Celsius. In these temperatures, the primers can bind to the two ends of the DNA strands. Once the primers knows exactly where it has to bind, it is like a GPS coordinator. But if the temperature is too high, then the primer will bounce off. If the temperature is too low, then it can bind at wrong places of the DNA where it is not supposed to. So maintaining the temperature is important. Then we have the next step, the final step, that is extension, in which the there comes the Taq polymerase. We have to increase the temperature to around 70 to 75 degrees Celsius. This is the peak optimal temperature at which the Taq polymerase works. The Taq polymerase was extracted from the bacteria Thermus aquaticus. It is found in the hot springs. That is the reason why it is heat resistant up to some extent. The Taq polymerase will pick up all the DNTPs, and then it will bind them to the 3' ends of the DNA strands. Once this process is done, the whole cycle is completed. The first cycle will be the most imperfect one. After second and third cycles, the DNA gets more and more perfect, and after almost 30 cycles, there will be more than a billion copies of only one DNA strand. Now I will be presenting the types of PCR. First is the reverse transcriptase PCR. The normal PCR would work in RNAs because the RNA is different from DNA. So a reverse transcriptase enzyme has to be used here. The reverse transcriptase enzyme will convert the RNA to complementary DNA. Once it is transformed to the complementary DNA, then the PCR can be done on it. That's why this is called the reverse transcriptase PCR. Then we have the nested PCR. In nested PCR, there will be 2 rounds of amplification. In the first round, the outer primers will bind to the DNA strand, and in the second round, the inner or the nested primers will bind to the DNA strand. This is more specific type. Then we have real-time PCR. If a patient is suffering from HIV and we cannot afford to wait for the cycle to complete, 30 cycles to complete, in real-time PCR, it gives an estimated estimation of the worsening or the betterment of the condition of the patient, and it gives the estimation by fluorescent emitting. Then we have quantitative PCR. This is directly linked to the real-time PCR. It, as the name suggests, it gives us the amount of DNA, the amount of complementary DNAs and RNAs present in the sample. Up next, we have race PCR. In the race stands for rapid amplifications of complementary DNA ends. It amplifies the terminals of the messenger RNAs. And at last but not the least, we have multiplex PCR. If a patient is suffering from respiratory distress and we have to test we have to add many primers without using the convenient conventional PCR, we can use multiple PCR where we can mix multiple primers. For example, for flu A, flu B, RSV or COVID-19, we can use multiple primers in one time, and that's why this is more efficient than PCR. From here on, my group mate will take it from here. Thank you and have a nice day.
Good morning, everyone. I'm Tanvi Kumar, roll number 143. Now that we have seen the principle and working of PCR, let's put light to its clinical applications. PCR has revolutionized modern medicine as it can amplify very extremely small genetic material very accurately. It is used in following cases: infectious disease diagnosis, genetic disorder detection, cancer diagnosis, forensic science, prenatal testing, and transplant medicine. Let's discuss each of the applications separately. PCR in infectious disease. PCR detects the genetic materials of disease-causing microorganisms with high accuracy. It enables early and accurate diagnosis even when only very small amount of genetic material is present in the sample. This helps the doctor to provide timely treatment, and PCR helps identify a wide range of infectious agents ranging from COVID-19 viruses to Plasmodium species malaria parasites. Rapid detection allows timely treatment, effective patient management, and better control of disease outbreak. Next, we have genetic disorder and cancer detection. PCR is widely used to detect inherited genetic mutations associated with various genetic disorders. It helps identify mutations responsible for diseases such as cystic fibrosis, sickle cell anemia, thalassemia, and Duchenne muscular dystrophy. In cancer diagnosis, PCR detects gene mutations, chromosomal abnormalities, and cancer-related biomarkers, enabling early diagnosis and personalized treatment. PCR is also used to monitor treatment response and detect minimal residual diseases, helping identify cancer recurrence after therapy. Next, we have prenatal and neonatal testing. PCR enables the early detection of inherited genetic disorders before or shortly after birth. During prenatal testing, fetal DNA obtained through procedures such as amniocentesis and chorionic villus sampling can be analyzed for genetic abnormalities. In neonatal testing, PCR helps identify congenital infections and certain inherited metabolic and genetic disorders, allowing prompt medical interventions. Early diagnosis facilitates timely treatment, appropriate genetic counseling, and improved long-term health outcomes. Next, we have forensic science. PCR amplifies minute and degraded DNA samples, making them suitable for forensic analysis. It is used to generate DNA profiles, DNA fingerprinting, from biological evidence such as blood, saliva, hair cells, skin cells, semen found in crime scenes. PCR helps identify suspects, exonerate innocent individuals, establish parentage, and identify missing persons and disaster victims. Because of its high sensitivity and accuracy, PCR plays a vital role in modern forensic investigations and the criminal justice system. Next, we have transplant medicine. PCR is used for HLA typing, human leukocyte antigen typing, to determine the compatibility between organ donors as recipients. It helps detect early signs of transplant rejection by monitoring the changes in the recipient's genetic and immune markers. PCR is also used to identify opportunistic viral infections such as cytomegalovirus and Epstein-Barr virus in immunosuppressed transplant recipients. By enabling rapid and accurate monitoring, PCR improves transplant success rates and supports long-term patient care. Next, we all know in 2020 we encountered a global pandemic, COVID-19. At that time, PCR not only became a known name in medical field, but also a very well-known name in household also. COVID-19 is caused by the SARS-CoV-2 virus. This virus is an RNA virus. So conventionally, PCR cannot detect it directly. Reverse transcriptase PCR converts viral RNA into complementary DNA before amplification. RT-PCR is considered the gold standard for diagnosing COVID-19 because of its high sensitivity and specificity. RT-PCR works on the following principles. The viral RNA is extracted from the patient sample. Reverse transcriptase enzyme converts RNA into complementary DNA. The complementary DNA is amplified using PCR. Fluorescent probes detect the amplified DNA in real time. Detection of fluorescence confirms the presence of SARS-CoV-2 RNA. This is the procedure of reverse transcriptase PCR. The sample collection is very well known to us as it was done during the COVID times very frequently at every airport, at every place to prevent the virus from spreading. Next, what we don't know was the isolation of viral RNA happening. The reverse transcriptase enzyme used to convert the viral RNA into complementary DNA. PCR amplification happening to that complementary DNA, and then fluorescent probes used to detect the presence of the infection. Next, moving towards the conclusion of my presentation, I would like to enumerate all the advantages of polymerase chain reaction. It is highly sensitive, highly specific. It provides rapid results, accurate and reliable. Wide range of applications, as I have mentioned before, and minimum sample requirement. Next, every coin has two sides. With pros comes cons. So without limitation, this presentation would be incomplete. So limitations of polymerase chain reactions are as follows. With high sensitivity, it also has a high risk of contamination. It requires prior sequence knowledge, cannot differentiate between live and dead organisms, expensive equipments and reagents. Technical expertise is required and possibility of false results is high if technical expertise is not provided. Next, to conclude the whole topic, polymerase chain reaction is rapid, sensitive and specific technique for amplifying DNA. It has revolutionized molecular biology by enabling accurate detection of genetic material from very small samples. During the COVID-19 pandemic, RT-PCR became the gold standard for detecting SARS-CoV-2, demonstrating the clinical importance of PCR technology. Continuous advancement in PCR continue to expand its application in diagnostic, research and personalized medicine.
ভালো হয়েছে, খুব ভালো হয়েছে, ভালো হয়েছে, ভেরি গুড প্রেজেন্টেশন।